Well, all right. This is a slide. So our today's speaker is Dmitry Grebенnikov, and the title of his presentation is Computational Methods for Multi-Scale Modeling of Virus Infections. Please, Dmitry, you can begin your lecture.
Yeah, thank you for the opportunity to give a talk on this seminar. And yes, I will be talking about the multi-scale modeling approach, which we are developing for studying the various virus infections. But mainly, I will focus on the computational methods, which are developed for these types of model. And actually, we can separate the types of mathematical models which are used in immunology into certain groups. For example, the models of systemic dynamics of the infections are usually represented in the differential equations, either ordinary ones or with delays, or can be also stochastic, which can be formulated with stochastic differential equations or via Markov chains. And by systemic, I mean the models which describe the infection in a whole organism, without the using the information about the spatial distribution of the processes. The next ones are compartmental models, which take into account that the infection takes place in various organs, usually, or tissues, which are called compartments. This is one type of ways to incorporate the spatial heterogeneity. Another one is the using the partial differential equations to model the variables distributed in time and in space. And the other type of models is a multi-scale model. And I will be talking about such a model. And it governs, it describes the processes which are at various scales, at the tissue level, at the organism level, and the intracellular processes. And actually, in these types of model, we use all the equations which are used for these other bullet points. And I will be talking about mainly as an example of the model of HIV infection, and this is a general scheme, mainly because it is in some sense easier to present, because the target organ, the target tissue, is the same as the tissue in which the immune responses are developed, which is the lymphoid tissue. And in this model, we represent this whole compartment of lymphoid tissues as one lymph node, which is an ellipsoid in 3D, and it also can have various, excuse me, and then also can have various sub areas in it. For example, the T cell zone, where the T cell type of immune response is developed, and the B cells follicles in which the humoral immune response is started. But we, in our model, currently only consider the T cell based immune response. And we consider two compartments: the compartment of the lymphoid tissue, which is this generalized lymph node, and the blood compartment. And there are three scales which are considered in this multi-scale model: the processes that occur at the tissue level or the level of the organ, the processes that occur inside the cell, and system level processes are mainly the transport of the variables between these compartments. And for HIV infection, there are cells in the lymph node and viruses and some other molecules, and these agents, they can interact somehow according to some logic at which the adaptive immune response is developed, and according to the logic of the infection spread. So various types of cells, for example, uninfected cells, CD40 cells are the targets for the HIV infection. The infected ones and the viruses, they can interact according to the schemes which are typically parameterized in the systemic types of models. But actually, we model these cells as separate units which can also move and interact. So we have to consider the model for cell motility, cell locomotion model, and some types of equations for modeling the virus, the diffusion of the extracellular viruses. Then inside the infected cells, there are also a number of processes, and these processes are governed by the models of virus life cycle, which represent the processes from the infection by the extracellular viruses. Then there are some intermediary products which are generated, and then later the new viruses are produced that can be secreted to the extracellular space. And the transport is modeled via some equations which I'll show a bit later. So the actual intracellular models are a whole separate subject, which and we have several models which are developed for various viruses. Here is the examples of the schemes of these processes for HIV, for SARS-CoV-2, and for LCMV viruses. And we developed the deterministic model, which are calibrated by the data available in literature, and then they are transformed from deterministic to the stochastic ones. And I'll describe later the methods which are used to model such stochastic models, either standalone or when it is incorporated in multi-scale model. But without incorporating them in multi-scale model, they also provide some insights valuable. For example, we can use the sensitivity analysis to predict which processes inside these detailed schemes produce the most influence on the number of viruses that can be generated by the infected cells. And so we predict the which processes or these reactants can be targeted by the therapeutic agents. And here is the example of the equations that can be used for the multi-compartment transport. These are general ODE equations. For example, for dynamics of number of infected and uninfected cells in blood, there is a source for the uninfected cell CD4 T cells in blood from which are generated from the lymphoid organs, and then there are some transfer rates and degradation rate, and these the right-hand sides of these equations depend on the number of cells which can leave the computational domain which represents the lymph node. And this number at current time is stochastic, and it is determined by the positions of the cell. So here we consider only the cells which are in the medular zone, so which are contacting this gamma M boundary of the computational domain. These are the cells that can leave the computational domain and can be transferred to the blood. And also for the dynamics of the number of variants in blood, we use the cumulative, the integral of the field of viral particles in the computational domain in these equations. And these equations, they can be considered as stochastic differential equations or random ODEs, but actually we use to model these types of multi-compartment transfer and other methods which are based on transforming these equations to the Markov chains in a similar way that is done in the for the intracellular models, and these methods will be described later. And I said that we have only one generalized lymph node, but actually we can consider more complex scenarios. For example, for the viruses in which the target tissue is different from the lymphoid tissue, or we can consider the development of the infection in various lymph nodes. And we can use the graph-based models for these types of compartment models, which are also developed by our group. And here is the graph for the human lymphatic system and for the lymphatic system of a mice, which is used for the multi-scale model of the LCMV infection, which is the infection used in experimental biology in mice. The next model is the model at the tissue level, the model of the immune cell locomotion. We use the following approach based on the equations, based on the second Newton's law, which can be written for each cell, which have its coordinate x, which have its certain radii and certain masses. And there are several types of forces that act on the cell. The first one is the intercellular interaction forces. So from the cells which are in contact with the cell E, we use the following parameterization using this cubic function, which describes the adhesion of two cells when they only contact each other up to a certain period, but when they if the distance between them is becoming smaller, then the repulsion forces are acting. And these forces are parameterized by the parameter of adhesion. So this is the parameter that is different for non-specific interacting cells and for specific ones. Then there is the dissipative forces, which are modeled as proportional by their magnitude to the velocity of the cell with the minus sign. And also there is the stochastic force, which we call the motility force, the force of inner motility. And we use various distributions to model these types of cells, this type of force, and it actually implicitly represents the structure of reticular networks which are which permeate the lymph node and which provide the guidance for the directions of the cells by which they can migrate. But we don't model these types of interaction explicitly. We model this by using some kind of these types of random distribution-based parameterizations to reproduce the experimental data. So here is we see that the motility force is updated actually at times delta t, which is 30 seconds for the immune cells, and we sample the magnitude of the force and the direction of the force. Magnitude is sampled from the normal distribution, and the direction is sampled in 3D based model from the von Mises-Fischer distribution, which is a distribution of the vectors, unit vectors on a sphere, and they are concentrated around the direction of the motility at the current time with some concentration parameter kappa. And the concentration of is linked with the amplitude using the following relationship, which allows us to model the experimental data and the fact that the cell can't turn at high speeds. When it is turning, the velocity is reduced. And to model such type of model, to numerically implement, we use the symplectic numerical schemes. For example, the first order semi-implicit Euler method, which is given on the slide. And this model was calibrated by the experimental data published in this paper, which provided the distributions of the following three metrics of the cells, which were captured during following a certain protocol during several hours at times delta t equal to 30 seconds. These metrics were captured, and these are translation velocity, the turning angle velocity, and the meandering index. And here is the distributions which were published in this paper, and here is the comparison of the these experimental-based cumulative distributions with the ones obtained by the model. You can see that we have a good fit for the translation speeds, but actually there are some discrepancies for the angle speeds and the meandering indices. And here is an example of the trajectories which are produced by the 2D model. And here is a number of trajectories in a whole lymph node. And we next used another paper on the spatial temporal redistribution of the lymphocytes following the infection of the mice by the ovalbumin. And here we can see that, for example, the difference in distributions of the distance of specific and unspecific cells toward the center of the lymph node. And we can see that after 12 hours of infection, the specific cells are moved towards the center of the lymph node, because these specific types of cells, they form the contacts with the dendritic cells, which are also located there. And we verified that we have a similar redistribution of this of the cells in our model when we initialize randomly the distribution of T cells and select some of them as specific and some of them as non-specific, and we place the dendritic cells inside the closer to the center of this domain. So this type of contact was actually reproduced by the model, and then we used the model by itself without integrating it to the multi-scale, but to answer the following question: what is the time? What are the conditions which are needed to be which we need to maintain in the lymph node so that the infection is controlled in the lymph node? And by controlled, we mean the following scenario. So if the lymph node under consideration is uninfected, and at time 0 an infected cell comes into the lymph node, and it can be different types of cell. For example, it can be dendritic cell, antigen presenting cell, which is non-motile, and it can be CD4 lymphocyte, which also moves. And we varied two types of parameters: the frequency, so the number of the specific CTL cells which need to find this infected cell up to a certain time, so that it doesn't have time to produce viruses. And the infected cell starts to produce viruses around 24 hours post-infection. From 18 hours, this process is started. So we can estimate that there should be about 5% of HIV-specific CTLs in a lymph node, so that they have time to find this these infected cell and prevent the propagation of the infection in this lymph node. And also we studied the effects of the reduced motility of the cells, which can be interpreted as implicitly, which can be interpreted as the scenario of fibrosis, in which the cell reduced their motility because of the collagen deposits, which are generated in large quantities in a lymph node in chronic infection. Then another type of transport which occurs in the lymph node is the transport of the not the cells, but other extracellular factors, for example, viruses and molecules, small ones. And we use the reaction diffusion equations for these types of to describe this type of their transport. And for example, the equation for viruses. And we can see that here is the source term, and I'll later describe the equation for this term. But actually, this term is moving in space, because the cells which are infected, they move. So we have moving sources, and also the amplitude, the number of this function also depends on the time. So it depends on the solution of the intracellular model. And for these types of model, we use the finite element method. For example, here is the weak formulation is described using the Crank-Nicolson discretization in time. And we use the for special approximation a certain time of this field, we use the finite element subspace or an unstructured tetrahedral mesh. And we use the meshes which adaptively refine to the sources of these viruses. And the sources are the areas occupied by the cells which are productively infected at this stage of the infection. Here's the example of the screenshot of the multi-scale model. So we have some cells which are moving, and we have also the fields of viruses and other molecules which can be studied. And next, there is an intracellular model which is solved at each infected, at each susceptible to the virus cell. And for the HIV, the susceptible target cells are CD4 positive T cells. And here is the scheme of this model. And so this consists of several stages. First, the extracellular viruses from the which number is governed by the solution of these extracellular equations. They can infect the cell, and there are some intermediary steps, and finally new viruses are produced which can be secreted to the extracellular space. And for HIV, it has the following stages. For example, the extracellular virus, it can bind to the receptors on the membrane of the cell. Then it exports its RNA to the cytoplasm, where it is reverse transcribed to the DNA, viral DNA, which can then be transferred to nucleus and integrated in the chromosome of the cell. And then this type, and at this stage, the cell is infected, but it can become either latently infected or, in some circumstances, productively infected. In which case, the following stages occur, and it consists of transcription of viral mRNA, then their export and translation, or synthesis of the proteins, either structural proteins from which should be inside the virus, or the regulatory processes, proteins, which actually regulate these the rates of these stages. For example, TAT is used to increase the transcription rate, so it is called the transactivation protein, and REV protein is used to it enhances the export of these mRNAs to cytoplasm and their splicing to different subcomponents. And then, so this regulation is used to increase after a certain time the production of these mRNAs, which then translate the components of the viruses, and then they are transferred to the membrane with viral RNA, and they can be assembled into pre-virion complexes, which then are budded and maturated finally. And we develop the deterministic ODE-based model with 24 variables and 55 parameters, which are estimated by the from literature. So there are some parameters which are like the basal transcription or translation rates, which are the same for all viruses, and some are virus specific. And also for the various viruses, the scheme differs. So some of them are not, for example, integrated in the nucleus. And we use the actually these stages are mainly linear and the variables are transformations from one stage to another, or synthesis of new variables, or the degradation or elimination of various parts. And for some processes which the nonlinear parameterizations, for example, to describe the effects of the regulatory proteins and the rate of the assembly of viruses from its subcomponents. And actually, we use this following parameterization in the latest models, where the rate of the assembly is proportional to the number of proteins, structural proteins, and viral RNA, but we limit the assembly rate by the least abundant protein using this type of multiplication of various Michaelis-Menten functions. And here is the solution of deterministic model for HIV life cycle, which for one cell which is infected with 4 viruses at time 0. You can see that there are some intermediate steps, and finally, about 24 hours post-infection, about 100 new particles is synthesized and can be secreted to the extracellular space. But we see here that here all the units of the variables, they are in the numbers, not the concentrations. So here is, for example, 1.5 number of DNA, viral DNA, which is integrated. But actually, it can only be discrete number 1, 2, 3, etc. And so this we transition from these deterministic continuous type of models to the stochastic based on Markov chain based models, because the deterministic ones, they are only valid at high concentrations, and it's when there are small numbers, then it can the differential equations can either produce some non-physical solutions, or they can just not capture all the scenarios that can happen in the cell. So how is the deterministic model transformed to the Markov chain? The variables of the intracellular models in Markov chain can have non-negative integer values. And here is an example of the first equations of the deterministic model and the last ones, and how they're transformed into this Markov chain table. So we list all the processes that can occur in the scheme, and for each we define the event which occurs by 1. For example, degradation of viruses, it happens with this propensity or intensity, and as a result, if it happens, then number of viruses is reduced by 1. Or for the binding rate, the extracellular is decreased and the bound viruses are increased, etc. And we use the already calibrated values from the deterministic model in these Markov chains. Formally, this stochastic model is defined by the following probability of events. We define the probability that if at time t the system is in state x, then the probability that event k from this table from the previous slide occurs in some infinitesimal interval is proportional to the intensity or propensity of this rate, ak. And if we define it in such way, then the dynamics of the stochastic model, which is discrete state and continuous time Markov chain, is governed by this type of equation. Where pk is the Poisson counting process for the kth event at the on the interval from 0 to t, and rk is the column vector from this stoichiometric matrix. So it is the vector which consists of zeros, ones, or -1 values, and it determines the state transition associated with this event. And we can see that the this is the Markov chain because the probabilities of the next stage are only dependent on the current propensities of the system, so which depend on the current solution, and it doesn't change between the transitions. Therefore, it is called the homogeneous Markov chains. And we can use various numerical methods, Monte-Carlo methods, to implement the stochastic realizations or trajectories of such for our model. And the easiest one are the direct method, which is known by Gillespie name, or the first reaction method, which is easier to describe, but is less efficient as it requires k number of pseudo-random variables at each stage. But it just samples the expectant time for each reaction and then chooses the reaction which is the first one to come. So tau is the minimum of this tau k, and the argument of this minimum is the reaction that will occur, and we increment the time to t plus tau and make the kth transition. For example, if k is 48, then we decrease a certain variable by 1. And here is the numerical solution of the stochastic model using these Monte-Carlo method to simulate a large number of trajectories. We can approximate the evolution of the distributions of the variables at certain time. So here it is described as a heat maps, and here is the first variable, and here's the last variable of the model, which is shown for brevity. And we can see that it also is this model is also initialized with 4 viruses, but at the end we have a heterogeneous distribution with which is multimodular, and we have various peaks. For example, the peak at 0, so the infection can actually become extinct and no viruses will be produced. Otherwise, it can be either around 80 or 150 or some different value. And this type of broad dynamics is not captured by the deterministic model. and we can actually predict these distributions at certain time, or the probability of productive infection. So, for example, the probability that there would be some non-zero number of proviruses or viral DNA at certain time as a function of the multiplicity of infection, which is the dose at which one single cell is infected. And we can see that these multimodular distribution of the number of viruses that the cell can secrete is actually caused by a certain distribution of the number of proviruses that can integrate into the chromosome of the cell. So if there would be one provirus, then it will correspond to this peak, and so on and so on. And we also have extinct realizations where no DNA is integrated. But if it is integrated, then there is also probability that up to a certain point there will be no viruses to produce, just from the stochastic nature of this process, and that the variables can only have discrete values. And also, actually, I'll skip this. We also can show that with various types of representing the processes of viral virion assembly, we can show that actually it can happen that the viruses which are assembled, they can be incomplete in their number of proteins. So the rate is, so this event is triggered, but there are no, the number of structural proteins which need to come to the virus is less than needed. And so we can have also predict the distribution of the number of these proteins in a virus. And next I come to the part of what happens with these intracellular models when they are integrated in a multi-scale model, and with other blocks of the multi-scale model. So, for example, the source functions in reaction diffusion equations for viruses is dependent on X and T, and is determined by the number of cells which are infected at time t, and by the positions of these cells at certain time, and by the number of viruses that can be secreted at this time, if which is the solution of the intracellular model. And on the other hand, we have the initial condition for the intracellular model is the number of cells that can infect the cell is determined by the number of virions which are in the extracellular field at the position of the cell at current time, and by the number of neighboring cells which can also transfer the viruses to the infected cell. And this means that in the Markov chain, which was homogeneous, it becomes time inhomogeneous, because the propensity, for example, the process 2, the process of binding of the extracellular viruses to the membrane, becomes time dependent. So this variable depends on the solution of the extracellular models, and also these K bound, the binding rate is actually also modeled as some exponentially decaying function with characteristic time td, which is due to downregulation of the receptors on the membrane of the infected cell after a certain time. So it is the process by which cell defends itself from being infected by too much variance. And because these propensities now depend on time, there are certain Monte-Carlo methods, different Monte-Carlo methods which need to be used to model these type of equations. And there are two categories of them. First, we use the integral-based methods, and to determine the time for the next reaction or the index of the next reaction, we need to solve these integral equations because propensities depend on time now. But now we actually use the uniformization methods, which are also known as thinning methods or method of maximum cross-section. And the idea of this method is the following. To the table of the marker chains, a certain virtual transition or event with index k+1 is added. And this virtual transition doesn't change the state of the system. And we define the uniformization rate as the upper bound of the sum of all the propensities which can be on a time horizon that we are interested in. So we can estimate this upper bound. And then we define the propensity of this extra transition in the following way, such that the total sum of the propensities in the extended system is constant and is equal to the uniformization rate. And this means that it doesn't depend on time. And this allows us to simulate this system. For example, it can be shown that the number of transition from 0 to t is distributed as Poisson random variable with parameter a bar times t. And we sample it from the Poisson distribution. And then the time points at which the transitions in this extended system will take place are distributed as the first order statistics of the uniform distribution. So it is uniformly distributed from 0 to t and ordered in such a way. And then if we know these transitions, then the actual sampling of the transition indices is done the same as in the direct method for the extended or uniformized system. And we use this approach. And here is the illustration for the just an illustration for the first part of the first variable of the intracellular variable, the number of bound viruses variants, given these time-dependent extracellular conditions. So we have changing in time number of viruses and the number of cells which are in contact now with the cell under consideration. And we see that the propensity rate of this binding intensity, binding rate, is dependent on time. And here are 5 trajectories of the solution for this cell with such extracellular conditions, which is simulated. And then in the whole multi-scale scheme, we then have a hierarchy of the time meshes from different blocks of this multi-scale model. So we have we have time step h, which is used for numerical integration of cell motility model and reaction diffusion equations. For cell motility, we also update at certain times delta t. We update the motility, the stochastic inner motility force, and then there are moments of the intracellular transitions which can take place in all the cells which are now either susceptible to infection or productively infected. So we and then we sample these moments, and they can occur. The time steps can be either less than h or more than h. It depends on the current stage of infection. And also at some times we can adapt the uniformization rate. And finally, the numerical methods for these stochastic Markov chain-based models, they can be computationally expensive if when the variables take values which are very large in the intracellular model, and they can be, for example, a certain number of viral proteins is around 1 million, and when these there are these high numbers, then it is not actually necessary to perform the stochastic to treat these processes stochastically. These are only useful for the small valued variables where the stochasticity takes place. So we use these approximate method, which involves taking trace of the indices of the variables which are larger than a certain threshold. We use 10 times 4 for this threshold, and which have values less than this threshold, they are treated with the stochastic model, and the other ones are integrated numerically. And this involves this allows us to actually use it for a multi-scale model where there can be many productively infected cells. Yeah, so that's basically it. So we have we can say that these intracellular models they should be treated as a stochastic discrete state continuous time Markov chains, and when they are because for small valued systems the results of the deterministic model are incomplete, and we cannot model the heterogeneity of the distribution of the number of viruses that can be secreted, and or that there would be an extinct infection. And then when these models are integrated in a multi-scale model, there are certain methods like uniformization methods which need to be implemented to stochastically simulate this multi-scale system. Thank you for your attention.
Yes, thank you, Dmitry. It was quite interesting to know about these models which you develop. It's quite complete and quite complex models. And traditional question when we talk about these individual-based models or hybrid models, in your simulations, what is the usual number of cells you consider?
Yeah, so for the multi-scale model, we only used it for to study certain questions about the dynamics of HIV infection when to study the local bursts of infection that occur in a lymph node when one or several small number of infected cells is migrated to this lymph node, and we only used these to simulate during around 1 day. So
no, yes, excuse me, I did not understand. So how many cells you consider?
Yeah, so it also depends on the computational domain that we consider. If we consider the whole lymph node, then there are about million cells.
Well, it's quite
quite
many. Yes.
Quite many cells, but we only studied it for like a small time span. So it's not, it was not used to model the course of infection during months, which is relevant for HIV.
But you say during 1 day, or what?
Yeah, about
so if
1-2 days.
If you consider 1 million cells and 1 day of physical time, so what is the computational time?
It's around several hours.
Several hours.
Yeah, but we need to, I mean, to use to investigate more scenarios to estimate so that I can give you more specific answers.
Yeah, but do you use parallel computing, or how do you do it?
Yeah, we also do parallel computation for such parts. For example, we can run in parallel the cell motility model and the diffusion model for the extracellular factors and the intracellular model. So we like consider parallelization by processes.
Not by cells, right?
Not by cells. Yeah, it is more difficult, but can also be done. Yeah.
Well, please, if there are questions in the audience, you can ask, and waiting for other questions I would like also to ask, because you mentioned, but maybe I just missed that, the transition between stochastic and continuous models. So if you have like a, for example, small number of variants and large number of variants, so do you consider this transition from stochastic to continuous, or you consider only one of them according to the situation?
You mean this high approximate method which we use, which so about this slide, yes?
Well, I'm not sure about the slide, but it seems to me, maybe I'm wrong, but it seems to me that you mentioned a possible transition, because, okay, let me put it more clear. If you have several variants in a cell, okay, you use stochastic method. If you have like millions of viruses in the extracellular matrix, you use continuous method. Do you have both of them at the same time in such a way that you have a transition between them from small numbers to large numbers, or you use only one of them, either stochastic or continuous?
Yeah, thanks. Yes, we actually we do not decide which one to use, but we use this hybrid method, and in this method we track at during the time the values of the intracellular variables, and we group them in two categories based on the either if they exceed or not the certain threshold. And
but it can change in time. So,
yes, it changes in time, and so the state space for the stochastically treated for the stochastic model is actually changes. It can be decreased or increased during the simulation, and of course it should be stated. It can be like this algorithm can be implemented on the one slide, but it actually
sorry, excuse me, but I did not understand. Let me ask again. In the beginning of simulations, you have 10 viruses in your computational domain. At the end of your simulation, you have 1 million of viruses. So how do you consider them, as stochastic, discrete and stochastic, or continuous variable?
Okay, let me show this slide. So here is the correspondence between the deterministic equations and the Markov chain-based equations. And we can consider either left one or the right one. But actually, we can make a hybrid method in which case we use these equations for the variables when they exceed a certain threshold, but use the Markov chain for the other variables which are still small valued.
No, you say for the other variables. No, my question is about the same variable, not for the other variable.
Okay, then for the same variable which started as small values but then exceeded this.
Yes, it's exactly my question. Yes.
Yes, then we treat them first from the start. We treat them as stochastic ones, but when they exceed the threshold, then we do not. Then we exclude them from this Markov chain, and we integrated them with the.
All right. Yes, now finally I understand. This was exactly my question. The transition for the same variable from a discrete model to a continuous model. So I suppose just if you have a discrete model with quite many particles, then you introduce a continuous variable as a concentration, as a number of particles in a unit volume. This is how you make this transition from a discrete variable to a continuous variable, right?
Yes.
Okay, good. I understand now. In the previous part of my questions, I forgot to ask you about the dimension. What is the dimension, space dimension you consider, 2D or 3D?
We use the 3D model, but for certain, now we use the 3D. But when we developed, we first started as a 2D model.
Well, I have also a question about the structure of your multi-scale model. In fact, I think all that is interesting, of course. But what is exactly the role of this blood compartment? Because in the beginning, one of the first slides, you showed the structure of your model with several compartments.
Yes.
This one. So for the blood compartment, okay, there is exchange of cells, but looks like this blood compartment is kind of passive. Nothing happens there. Then why to use it?
Yeah, you're absolutely right. In this type of model, it is just passive, because for the HIV infection, we consider that the most of the interactions occur in the lymph node, and we use the blood compartment just as to compare it with the experimental data which can be observed in blood. So
to measure some.
Here we do not consider any processes in blood except for the degradation rates, so they can also die. in blood, but we do not consider interactions, but they can be.
Also, you mentioned in the beginning, I think that you consider in your model like one lymph node, but if you consider several or the whole system, as you showed on some of your slides, then you can probably observe how viruses move from one lymph node to another one, right? Through blood, or maybe through lymph node, through lymphatic system.
Yeah.
By the way, how do they move? So is it possible to? Probably not. It's probably lymphatic system how they are transported from one lymph node to another one, right? Not blood.
Yeah. So actually, this is also a topic which we are developing as a separate model now, which is the multi-compartment models of transfer of cells in the lymphatic system, which is also how to say, which is connected with the blood system. I didn't describe it here, but actually the interaction between lymph node, between the lymphatic system and blood is happening in each lymph node. So each lymph node is permeated by the blood vessels, and there can be a certain exchange in them. And finally, the lymphatic system, it just collects the lymph, and here is the output vertices which correspond to the vein, to the jugular vein. So yeah, we're also developing these multi-compartment models, but haven't used them yet in multi-scale context.
Yeah, this becomes very, very complex. And by the way, about this complexity, when you consider intracellular model, you mentioned 24 variables, 51 parameters. Do you really use? Do you really need this complexity? Because, well, in fact, it can be summarized in a very simple way. The virus comes in, enters in, and then goes out.
Yeah.
So, do you need all these many variables and parameters to describe this basically in the macro scale, just one stage or two stage model, just in and out?
Yeah, that's a very good question. We developed these detailed models because first they are of interest by them, by themselves due to sensitivity analysis. We can predict which targets do not have yet the therapeutic treatments, but we used it actually for HIV, but in a very simple way. But we included also the intracellular regulation of the interferon response, and we have models which are actually extended about the virus life cycles and how it corresponds with the intracellular interferon-based defense system. So, and in multi-scale context, it could be also interesting to study the distribution of the drugs and what is the bioavailability using this type of multi-scale models.
Yeah, I see. Yes, with interferon, it becomes really, well, it makes sense because we know that infected cells produce interferons, they go to surrounding cells, they can protect them, and so on. It becomes really even more complex, and it's not just a linear direct process, it's more complex. All right. I think that I have a couple of more questions. Moreover, there are no other questions from the audience at the moment. So at some point you mentioned that cells, when they are infected, they can be productively infected or latently infected. So, so it depends on what infect. this choice between these two?
Yeah, that's a very good question, but I just mentioned it for like because it is, but we do not model it. We just use only we consider that a cell can only become productively infected now.
Yeah, but independently even of your models. So do you have some ideas, some understanding? It depends on what, I mean.
It's actually quite complex and it is not very clear for me. But there are certain activation factors for this type, but it is not. I didn't study it well, and I can't actually say anything specific.
Is it possible that some drug treatment, medical treatment, just kills productively infected cells, but latently infected cells remain alive? Is it possible that it acts like that?
Yeah. It is.
Well, I think that, well, my last question, maybe I also missed that, it's slide number 10.
Yeah.
Well, as far as I understand, but I'm not sure that my understanding was correct, you mentioned that there is a non-uniform cell distribution here, and some cells move towards the center, and something like that. Is it really so? And if it is so, how you describe, because you did not mention chemotaxis here in your model, then how do you describe this directed motion and non-uniform cell distribution?
Yeah, thank you. So we can see here that there are, for example, this plot is the, or this one is the velocity of the non-specific cells and specific cells. Specific cells are the ones which come in contact with the dendritic cells which are in the center. So we do not model explicitly any chemotaxis. How is it possible that they are redistributed? In our model, it is a result just only of the fact that the strength of adhesion for specifically contacting cells is 10 times more than for the non-contact cells. And so this means that cell just like randomly migrate, scan the lymph node, but when they encounter the DC, they form a prolonged contact, prolonged stable contact, and therefore they decrease their velocity, and they are mainly stay in the center of the lymph node.
Yeah, because the dendritic cells are basically at the center, right?
Yeah, they are initialized in the center, and they barely move.
All right, thanks, I understand that. All right, thank you very much. And since I don't see other questions from the audience, then we finish our today's seminar. Thank you for your interesting presentation. Thanks to all our listeners. Have a good day. Goodbye.
Goodbye.
Thank you very much. Goodbye.